Alright, this is going to be a quick video about insulin production using bacteria. So you have a double-stranded vector and you have a promoter sequence. You cut it here with the restriction nuclease, and then you insert the DNA coding sequence. So this would be a insulin gene, and then you put that into bacteria, and then it's gonna produce the expressed protein because of the promoter. And there is a Shine-Delgarno sequence is needed in bacteria if using bacterial cells. So bacterial cells need the Shine-Delgarno sequence, and that's specific for bacteria. So you won't get it from the eukaryotes. You have to synthesize that specific sequence. Yeah, and it's better to use prokaryotes because they're easier to handle with. Okay, and this is describing this over here. So insulin in humans is derived from a polypeptide called proinsulin. So proinsulin is this thing over here. So this part has to be cut out so that this thing the this H's bind over here, and then these no. So these S's bind to each other, the hydrogens get kicked out, and then this one goes and binds to that one, and then you get insulin. Then you can reverse it, can cut the disulfide bonds and go back there. So this is what happens normally in our body. Proinsulin folds itself by specific disulfide bridge followed by proteolytic process. We talked about that. Bacteria are unable to process human proinsulin, therefore both chains are separated. Chains are separately produced in bacteria. So you have a chain and a B chain. So let's say this is the A chain, that's a B chain. So that's produced, and then that's produced because you have the vector with the A chain, vector with the B chain. Put it into the bacteria. It's going to make some insulin, and then you just let the A chain and B chain react with each other. You get insulin. So pretty separate upon mixing the two chains and subsequent oxidation to form disulfide bonds. Active human insulin is generated. So active human insulin. So the keys, one of the things to remember is oxidation. Oxidation leads to disulfide bonds.